Okay, welcome to Sophie's fancy screencastify presentation. That is a very new concept for her that you can create these sort of things. So please excuse me if there are some sort of technical difficulties or a little bit of awkward parts during this video. Also, please excuse my hair because I am very tired right now. And so I created this presentation in a response to basically my presentation that I gave on Monday because I feel that I did not make it clear about the mechanisms of plant virus transmission through aphids and overall all phloem feeding insects. And I feel that if I reiterate how the viruses are transmitted through insects and other through aphids and other insects, then it would help you understand the science of my research more and also help you understand this important ecological relationship as a whole, which I find incredibly interesting. So I'm gonna enter full screen. So first, here's the question that I thought was facing everyone up: how are plant viruses transmitted through aphids? And this slideshow and this question can be addressed to pretty much all phloem feeding insects, but I'm just going to use aphids because that's what I'm most familiar with. So, for a bit of review, remember plants are mobile organisms. Therefore, all functions involving contact, they must have assistance from other organisms such as insects, and they have a mutualistic relationship with insects that we learned about in the bee movie, which was a really fun movie. And so the bees basically would pollinate the plants, and the plants would have access to sexual reproduction this way, but then the bees would also get food to make their honey. And it wasn't quite as fancy as it is in that photo, but it was something similar. And so here's the plant-insect virus relationship. Plant viruses have basically hijacked the plant-insect relationship to their own advantage. About 75% of plant viruses are insect transmitted, and aphids transmit about 50% of these insect transmitted viruses. So there are other insects such as thrips, which can transmit plant viruses, but aphids are the main one. Therefore, viruses must alter the entire plant-insect relationship. They decrease plant defenses, such as they activate this salicylate pathway, which leads to decreased plant chewing resistance. And also they increase performance and fecundity of the vector. And basically through decreasing plant defenses and increasing the performance and the fecundity of the vector, the vector does better on the plant, and then the vector is more able to transmit the virus. And so viral transmission with insect vectors is only with, or primarily with, phloem feeding insects. I've only heard of it with phloem feeding insects. And so viruses are found in the phloem, or the other vascular tissue. And the two methods of transmission are circulative and non-circulative. But for all methods of transmission, it starts with the attraction. And I talked about how there's certain things such as coloring or smell, etc., that will attract the insect to the infected plant, which is a funky yellow color. And then it'll settle and feed on the plant. And when it feeds, it uptakes the virus. Then it'll leave the plant and go to other plants and then feed on those plants. And when it feeds on those plants, then it'll transmit the virus. The two methods of transmission are circulative and non-circulative, which I will go into next. So first are circulative viruses. Circulative viruses remain in the mouth parts of the vector or the aphid, and I really like this diagram because it has a little bit of shading or color codeness. And so basically with circulative viruses, they are only viable for a short time of a few minutes, sometimes even only seconds, because they remain in only the mouth parts. And the TUMV virus is one such virus. So as you can see, it just remains right here in this little shaded region. Circulative viruses enter the insect's circulatory system and are viable for a long time, sometimes even for the entire insect's life. And Additionally, there are two types of circulator viruses, non-propagative and propagative. So with a non-propagative virus, basically what will happen is the virus will enter the food canal. It'll go into the midgut, and then the hindgut and near the hemicole, and then it'll go back to the salivary glands and back to the plant via the saliva canal when the aphid feeds on another plant. So in the propagative and the non-propagative viruses, circulated viruses, the virus will go through this path. And with the non-propagative virus, the virus will just hang out in the circulatory system. With the propagative virus, the virus will actually infect host cells and replicate, which is significant. Basically means that not only is it a plant virus, it is also technically an insect virus because it is infecting the insect, which I find to be super cool. Okay, so I'm going to bring us back to these fancy slides that I'm really proud of, but we're going to look at them from a new perspective. So both the circulative and non-circulative viruses attract insects. But then there are two pathways that, and the past sciences have kind of attributed the circulative viruses to one and the non-circulative to the other, at least in the literature that I've seen. So with the circulative, it causes increased growth and fecundity of the aphid, or increased performance, which leads to a better host and crowding, which makes sense because the virus transmission is viable for a long time. With the non-circulative, it would make an unsuitable host, and therefore the aphid will leave quickly and go transmit the virus while the virus is still viable. And this is what makes sense logically if we think about it. However, we have found evidence, or at least the scientists have found evidence actually in the Castile lab, that the non-circulated viruses such as the TUMV actually do the same thing as the circulated viruses, and vice versa. And maybe one virus might be able to do both, et cetera, et cetera, et cetera. And so that is the main research question, is can a virus promote both types of viral transmission? Is there a really difference in a viral effect on plants? And in fact, one of the ultimate questions that I've been considering, and some people that I've talked to casually in the lab have been considering, is if the virus is intelligently promoting this increased aphid growth and fecundity or increased aphid performance, or is this just an indirect cause of infection? For example, if we damaged a plant in any sort of way, would it result in similar effects? Whereas if we damaged, it's just basically we're wondering if is the virus actually adapting for this intentionally. We're basically asking about the intelligence of the virus, but that's not that important. I just find it to be super interesting, and I've become rather obsessed with viruses. And here are some references if you feel like checking some papers out. And that's pretty much it. And I hope you enjoyed this, and I hope things are a little bit cleared up.